It's time to discuss Science Practice 5, which is statistical tests and data analysis. So, as you can see, part of that is a lot of the math calculations we've been doing in the class. For example, performing the chi-square analysis, calculating rates and means and ratios and percentages and things like that. Now, I did see on the College Board website this, which says that the 2020 exams don't require a calculator. You should be able to just do the calculations by hand. So I guess you won't be expected to do things like a chi-square. Now, a 4-function calculator like you would just have on your phone is a reasonable expectation. So percentages and means and stuff are okay. But another part of that science practice is to be able to use confidence intervals and or error bars, okay, or understand standard error to compare means. All right, so some of our task verbs for this are calculate or determine or evaluate. So let's go ahead and look at question 4 from the 2018 exam. They give us a graph, and you can see that the graph has error bars on it. And what we're looking at is survival rates for bed bugs. Okay, and they've gone ahead and knocked out some genes from certain strains of this bed bug species. So we've got strain 1 that has all the genes intact. Okay, and then the negatives indicate which gene they've knocked out, and they're just looking at how that affects their survival when they're treated with a certain insecticide. And the question says, identify the control strain. Okay. We're trying to identify the control strain and then use the means and confidence intervals in the figure to justify the claim that ABC8, okay, the gene that they're looking at up here, is effective at providing resistance to beta-cyfluorothin, which is the insecticide they're using. So what's our control? The control is the one that they haven't changed. So we would say like, well, strain 1, right? They haven't knocked out any genes. So strain 1 is our control. Now, it's just the task verb identify. So strain 1 is the control. That's your answer. And then the control is the one that we compare others to. So it's saying we're going to use that information and the information in the graph to justify the claim that ABC8 is effective at providing the resistance. Okay, so it's telling us these are the 95% confidence intervals for each of these sets of data. And a 95% confidence interval means that our true mean, okay, it looks at the variability in the data and it gives us the probability that the true mean is somewhere within those lines. Okay, so we're 95% sure. Okay, this is the mean we got for our samples, but in the actual whole population everywhere, the mean is somewhere between here and here. Okay, we're 95% sure of that. So if you have data that's a little more variable, you might have a larger error bar or confidence interval on that. But what we want to look at is this one and this one, where they knocked out the ABC8 gene. Okay, and what we want to do is compare that to our control, because that's why we run a controlled experiment, to have a comparison. So what we see here is that not only are the bars shorter, but the error bars, sorry, these aren't really straight lines. The error bars are well below the error bars for our control. So the error bars do not overlap, and that shows us that we have a significant difference between the two groups. Okay, so you could say ABC8 is effective at providing resistance to the insecticide because when compared to the 95% confidence intervals for the mean or for the control group, okay, they have no overlap or they show a significant difference. All right. So you can see here using those confidence intervals and error bars to determine whether sample means are statistically different or not. And that is how that's done.